Hi, my name is Caitlin Woolley, and I will be presenting on behalf of the Damaser lab from the Lerner Research Institute at the Cleveland Clinic. Our abstract, Nanoparticles for Delivery of Elastogenic Factors to Treat Pelvic Organ Prolapse. I would like to thank ICS for the opportunity to present at this year's conference, and we have no affiliations to disclose. Pelvic organ prolapse is the downward descent of one or more pelvic organs, causing discomfort and reducing quality of life for many women. Below, you can see a visual of various pelvic organs prolapsing in females. And the primary risk factor we are interested in is vaginal delivery. When a female undergoes labor, the pelvic floor tissues undergo extensive remodeling, which later on in life can lead to prolapse. Current treatment options for prolapse are a pessary and a surgical mesh. Both provide mechanical support, as you can see here. However, there are no current treatment options that regenerate the damaged pelvic floor tissues. Previous studies have shown abnormal elastic matrix homeostasis is found in both human and animal models postpartum. To the right here, you can see the development of the elastin matrix, which provides structural integrity and elasticity, making it a critical component to the pelvic floor tissues. Now, Lysyl oxidase and fibrillin are important cross-linking and structural proteins on the elastin matrix. And when the tissue is damaged, for example during prolapse or vaginal delivery, matrix metalloproteinase will inhibit the cross-linking of LOX, weakening the elastin matrix. And when this happens, tissue inhibitor of metalloproteinase will inhibit the binding of MMP to LOX. So, our lab is focusing mostly on nanoparticles for a possible treatment option for prolapse. Now, these nanoparticles can be delivered to repair elastic matrix through controlled release of elastogenic factors via direct injection to the vaginal wall. The nanoparticles we use are polylactic coglycolic acid nanoparticles, which provide low toxicity with a controlled release of an elastogenic factor to the targeted tissues. The elastogenic factors we use are transforming growth factor beta-1, which promotes the LOX-mediated cross-linking. As I talked about before, LOX cross-linking is very important to the structural integrity of the elastic matrix and pelvic floor tissues. And then we also look at doxycycline, which augments elastic fiber repair. For testing our nanoparticles in vitro, we use Lysyl oxidase-like 1 knockout mouse model. So LOXL1 is an elastic matrix cross-linking protein, and it has been seen that the absence of LOXL1 protein in mice has resulted in prolapse development in a pattern similar to humans. On the right here, you can see our grading system for vaginal prolapse in these mice. The increase in grade increases with the severity of the prolapsed area. As you can see here, I am pointing towards, and the black arrow points towards the bulging. So non-epithelial vaginal cells are dissected from the wall of these mice. Because these are smooth muscle and fibroblast cells, they produce elastin and collagen, making it important to use in our studies. So our hypothesis is the use of TGF-B1 loaded nanoparticles and exogenous DOX can upregulate pro-elastogenic protein expression. The aim of our study is to treat non-epithelial vaginal cells from LOXL1 knockout mice with TGF-B1 loaded nanoparticles and exogenous DOX to measure relative protein expression for elastin homeostasis. Our in vitro methods are NEVCs are dissected from 9 nulliparous mice, 9 single delivery non-prolapse mice, and 1 mulliparous mouse seated on a 6-well plate for 21 days. And then these are treated with either TGF-B1 nanoparticles or exogenous DOX. And then Western blot analysis is used to analyze the following elastin matrix proteins. So our results showed that delivery increases expression of proteolytic proteins in LOXL knockout mice. As you can see here, MMP increases. And because of this increase in MMP, we see a decrease in proelastogenic proteins such as LOX and TIMP. And then when these cells are treated with TGF-B1 nanoparticles, we see an increase in pro-elastogenic proteins such as LOX, and then we see a decrease in proteolytic proteins such as MMP. So this suggests that these nanoparticles have pro-elastogenic effects because they decrease the MMP characteristics that inhibit LOX cross-linking. And then when these cells are treated with exogenous DOX, we see an increased expression of pro-elastogenic protein TIMP2. And then we see a decrease in pro-elastogenic protein LOX in fibulin 5 at various concentrations of DOX. And then finally, using exogenous DOX, we saw a decrease in expression of proteolytic protein MMP and the ratio between MMP2 and TIMP2. To conclude, TGF-B1 and DOX have pro-elastogenic effects in vitro, suggesting the ability to repair the damaged elastic matrix in prolapse. And the future direction of our work is to test DOX nanoparticles in vitro to see elastin protein expression, and then to test TGF-B1 and DOX nanoparticles in vivo with the Lysyl knockout mouse model, and then finally to use TGF-B1 or DOX nanoparticles as a minimally invasive delivery procedure into the vaginal wall to repair the damaged elastin matrix caused by either vaginal delivery or prolapse. And thank you very much for listening, and if you have any additional questions, please contact Dr. Damaser.